Hi everybody, my name is Clement Chan, and for my Chem 6BL at-home assignment, I wanted to do my experiment on the using purple cabbage as a pH indicator experiment. So to begin, I wanted to start with some vocabulary. Acids are generally the proton donators, and they usually taste sour, and then bases are usually the proton acceptors, and they generally feel slippery. pH indicators are used to help determine whether unknown solutions are acidic, basic, or neutral. There are many different types of pH indicators such as litmus paper, but in this experiment we will be using purple cabbage. Purple cabbage can be used as an indicator because they contain molecules called flavin, which are anthocyanins, and they are water soluble and they will change color depending on the pH of the solution that they are in. So in this case, the pH, if the pH is low and the solution is more acidic, the solution will become more red and orange, and in neutral solutions they will appear more purple, and in basic solutions they will appear more blue and green.
And yellow.
So for this experiment, the materials that we need are red cabbage, some water. I have DI water here. And then you can use any type of household products that you wanted to test for whether they are acidic, basic, or neutral. So I have some lemons for citric acid. I have some antacids. I have bleach, vinegar, Gatorade, and I also have some materials to help me out. So I have a measuring cup, some clear cups so that I can actually see the color change, and some filter paper. It will also be very useful and necessary to use some protective equipment such as gloves and eyewear, since some of these solutions are very irritating towards the skin. So
Baking soda was also used in the experiment.
So for the first part of the experiment, we will be cutting our cabbage.
After cutting the cabbage pieces into smaller pieces, it is then placed into a blender with DI water. This is to get the anthocyanin out because they are soluble in water.
So now that once the mixture is about into a homogeneous solution, you can then proceed with the next step. Okay, so now that you have a homogeneous pH indicator mixture, what you want to do is you get a filter and then you use a coffee filter paper. This is used to help take out the remaining cabbage pieces that are still in the solution. So next I'm going to pour it . Now that we have our purple pH indicator, we can use it to test our solutions. So in the cups down here, I have each solution labeled. So what I'm going to do next is use my indicator here as labeled, and I'm going to use this to transfer the pH indicator into each one and to see whether it's acidic, basic, or neutral. So what I'm going to do next is I'm going to transfer the pH indicator first into the vinegar. As we can see, this turned red. Once again, to compare it to the pH indicator, which is purple, This red indicates that it is an acidic solution. Next, we have our lemon juice. I'm going to transfer the same amount. Once again, we see that it is relatively red. Next, we have our Gatorade. So this red means that this is acidic as well. Next, we have our Gatorade. So the Gatorade didn't really change color. So it was red and it still appears red, so it should be acidic. Next, we have DI water. DI water with the pH indicator is purple. So, as stated, since it is neutral, it is appearing as purple. We now have our antacid. So this is sodium bicarbonate. So after it is appearing a bit purple.
So this means that it is a weaker base because it is still lighter than the original dark purple pH. Now onto the bleach. As I add the pH indicator, we can see that it at first turns a bit yellowish, but after it settles down, the camera doesn't really pick it up, but it turns a little greenish yellow.
Next, we will be testing baking soda. In the baking soda, we can see that it turns blue. This means that it is a basic solution.
Why is pH so important? pH is important because it is used in a lot of everyday life. For example, in the field of agriculture and gardening, soil is a complex system that involves many different factors that are affected by soil pH, such as microbial activity. Plants also have an optimal pH where they can thrive, so pH is very important in plant growth. Next, aquatic life is very regulated with pH as well. Water that has pH that is too high or too low can be harmful for the fish and aquatic life. Water treatment is also very regulated with pH. Next, there's the food industry where pH is measured to test for quality and the microbial activity in the food. It is also used to control the taste and other properties such as prolonging shelf life. So from this, we can see that pH is very important in our life, and it is very important that we understand what is acidic and basic. Next, a question that we should go over is why exactly is there a color change between the pH indicator in the acidic solution, neutral solution, and the basic solution? So to answer this, it is because in different pH conditions, the hydroxyl and or the methyl ether group attached to the carbon ring on its structure will undergo reversible structural transformations. At acidic conditions, the fluorylum cation is the predominant species and contributes to the orange and reddish colors. Due to the conjugation with the electron withdrawing beryllium ring, the phenolic OH groups of the fluorylum ion at C4, C5, and C7 are fairly acidic. At neutral pH, the neutral quinoid structure occurs. However, as the pH increases, kinetic and thermodynamic competition occurs between the hydration reaction and the fluorylum cation and the transfer reactions related to its acidic hydroxyl group. A second proton loss from the C4 hydroxyl group causes the anionic base with maximized electron delocalization over the three rings. Thus, as the pH increases and more protons are lost, the conjugation increases. In 6BL, we learned that increased conjugation means a decrease between the HOMO and LUMO orbital gaps. Thus, less energy according to this equation, or a higher wavelength is absorbed. This results in a higher wavelength of light that gets reflected or emitted. This is why we see the higher wavelength yellow-greenish color in basic solutions.